Hi, everyone. So today I'm going to talk about this research agenda that we have on the effects of education on health and SES. I've been working with Patrick and Leandro Carvalho, who's at USC, on these papers. We have, just so you know, we kind of like a lot of the stuff I'm going to be talking today come from like two papers that we already have. One is NBER working paper. The other one came out at PNAS last year. And then we also have some current work where we're looking at like education and socioeconomic status. In all this work, we're going to use like UK Biobank data, and we're going to use also a natural experiment from the UK to get at the causal effects of education. So why education and health, right? Education and genetic makeup have been shown to be associated with later life health. There's a large literature in economics that show that there's very robust association between education and health, and a growing literature trying to see how much of that association is actually causal, right? The education is actually causing better health. Some of the mechanisms that people think when looking at the effects of education are that more educated people have higher incomes, so they might be able to afford better care or better, healthier lifestyle. More education might allow you to better process health information and change your health behaviors, for example. Or more educated people might have like more self-control and be able to invest more in health than less educated people. So all that kind of just to think about why education might affect health. There's also some papers that suggest that health may depend on the interaction between education and genetics, but as among others, Matt talked a bit on Monday, there's actually lacking good empirical evidence for this type of G by E research. A lot of the existing work has been plagued by low power and endogeneity issues. So the question we're gonna be trying to answer is, does genetic makeup moderate the effects of education on health? Or Or similarly, does education affect the association between genetic makeup and health? To be more specific, we're gonna look at compulsory school age reform in the UK, and we're going to ask whether an additional year of school at age 15 brought by this reform affects obesity, lung function, and blood pressure in middle age. And then we're also going to ask whether this effect vary with polygenic scores for BMI and educational attainment. Now this doesn't work. Or this. Interesting. Okay. So you've talked about this in several lectures, but just to recap, there are some obstacles to doing this kind of G by E research in the social sciences. One is low power replicability. Most of these behavioral traits are polygenic, meaning that most of genetic effects are very small. If you look at then interactions effects are even smaller than these direct effects. So you really need like large samples to get at this even smaller effects. Also, most previous studies focused on candidate genes. Again, Matt showed us that a lot of those did not replicate because they were underpowered. So as a result, many of these results aren't really credible results. Another obstacle are endogenous measures of environment. So by endogenous, I mean measures that are plagued by omitted variable or reverse causality. So in our context, for example, health might affect educational attainment. Health in childhood might affect educational attainment. So that's an example of reverse causality. Or self-control might drive both schooling and health decisions. So that would be an example of omitted variable. And if the environment proxies for genetics, then the G by E estimates we might actually be picking up G by G effects. So that's kind of all problematic when we're just using endogenous measures. So what do we do then? We have these obstacles, we have this literature where there's enormous evidence, enormous interest, but very little credible evidence. So we thought, okay, so what can we do that would, further this line of research? So what we do in our paper is to combine a natural experiment to get at some of these endogeneity problems with polygenic scores. So the polygenic scores will add statistical powers when compared to the candidate team. So we use polygenic scores for a large sample, the large UKB sample, which will help us with power. The natural experiment is, as I mentioned, a well-known compulsory schooling reform in the UK, the 1972 UK raising of school leaving age. And that will help us with confounding factors, issues of reverse causality and omitted variable bias. Important, we pre-registered all an analysis plan, a pretty comprehensive analysis plan, where we described the constructions of all the variables and all the specifications that we were planning to use before the full genetic data was available. So the way the UKB released the data, the genetic data, is that we released for the fourth of the sample, and then they release for the other 3/4. So we were, we were happy that we had the opportunity to pre-register this analysis plan. I don't know what's going on with this. Let's see this. Okay, I'll keep trying. So I just wanted to give, to make an aside on the importance of pre-registering an analysis plan in this context, right? So as you saw in several lectures, there's been a lot of problems replicating G/E effects. Matt showed those pretty depressing graphs on Monday, where a lot of these results are just not real, right? So in this context, pre-registering, I would say it's a pretty important step. There are many degrees of freedom on the specification choices that you can make, and people in this area are pretty, maybe rightly, worried that people will be fishing for significant results. I don't know, I'm plagued by the gods of technology, I don't know. So what we did is that we kind of did power calculations based on the fourth of on preliminary results using a fourth of the sample, and then we kind of guided our specification choices on that. So in one of the papers that I mentioned in the beginning, we look at kind of distributional effects of education and health in this population. So we're asking where in the distribution of continuous health outcomes are the effects of education largest. And in that paper, we find that the effects are actually pretty concentrated. Instead of education shifting the whole distribution of health, it actually had effects that are much larger at particular points of the distribution and then others. So then we realize that, okay, if we focus on those points where the main effects are larger, we'll be better powered to find G/E effects. So we kind of, then based on our previous work, we decided to focus on these points, and we pre-registered the fact that we were going to do so that it wouldn't look fishy afterwards. So a lot of the results that I'm going to show you today are effects based on these thresholds where we saw the largest direct effects. So just saying, this kind of increased the credibility of the results and helped during the publication process. So just very quickly, we use data from the UK Biobank. This is, a very large data set. It has 500,000 individuals ages 40 to 69. The data was collected 34 to 38 years after the reform we were going to study. So all the estimates that I'm going to show you today are effects of education later in life, right? Secondary, like we have something that affected secondary education when the participants were teenagers, and we're going to ask how does their health, like 30 years later, when they're about like 50, 55 years old, are affected, are different, right, because of that reform. The great thing about the UK Biobank is that it has a lot of objective health measures. Participants went into assessment centers where nurses took their blood pressure, spirometry, anthropometric measures. And that's great in this context because it's been shown that socioeconomic status affects how people self-report their health. So in this case, when we're interested in the question, how does education affect health, we're interested in the question, how does education affect real health, not like self-reported, right? So using these objective health measures are great because of that. They also have genotypic data for the full sample. One drawback is that the data is not nationally representative. In particular, it's been shown that the participants are more educated than the general population. However, I'll show you that we have evidence that in our experiment we have internal validity, if not external validity. In our analysis, we're going to restrict the sample to participants of European ancestry because, as you know, these are the people for which the polygenic scores are most relevant to. We're going to restrict to people born in England, Scotland, or Wales because that's where the educational reform we'll be looking at took place. And we're going to look at people that were born between September 1st, 1947 and August 31st, 1967. Our reform cohort was born from September 1st, 1957 on. So this range of dates are supposed to get people that are born within 10 years of the reform cutoff. And hopefully this is going to get clearer when I show you some graphs. So how do we define, how do we code people from European ancestry only? We pick those ones who self-identify as white British and those whose first principal component of the genetic matrix is less or equal to 0. So what is this reform that we're going to use to get exogenous variation on education? So in 1972, England, Scotland, and Wales raised their minimum school leaving age from age 15 to 16. Importantly, this new policy only affected students born after September 1st, 57. So students born on August 31st, 57 or before could drop at age 15, and students born after that had to stay in school until age 16. So as a result, students born within days of each other who would otherwise had similar health and education ended up with 1 year difference in schooling. So that's kind of what we're going to use to then find what the effect of this extra year of education is on health. Yes?
Are there gradients within years across months in some of your outcome measures?
Not in the outcome measures, but in the education measures. Like there's this interesting, I don't think we have it in here because we're only using year of birth in years. But if I break this year of birth by months, there is this cyclicality because in fact, when people are born during the summer, they can still drop out at age 15 even after the reform, because they will be 16 by the next academic year. So there is some cyclicality on education, and in all our models, we're going to control for month of birth. So any cyclicality is going to be washed away by this dummies. Yeah. So, oh my, okay. So this is just a graph that shows you year of birth, fraction staying in school until age 16 by year of birth. And you can see that it was kind of going up even before the reform took place. But then after the reform took place, there is like this discontinuous jump, right? There are a fraction of students born here that were leaving school at age 15. And for people who were born after this date, they no longer could do that, right? So we estimate that there's about a 14 percentage point increase, discontinuous increase, in the fraction of students staying in school until age 16 at the policy cutoff. So how do we then kind of use this variation in education to understand whether there is a causal effect of education on health? So we're going to use this in a regression discontinuity design, which economists in this room probably know about, where we basically say, okay, so we have this discontinuity. This discontinuity is an exogenous source of variation in education. That is a source of variation in education that was not chosen by the individuals, right? So we can use that to study the causal effects of education on later life health. So we're basically going to ask whether the discontinuity in education that we saw generated discontinuities in health at the policy thresholds, and whether these discontinuities are different by genetic makeup. You have a question? No. Okay. So the identifying assumption of this sort of method is that people on either side of the threshold are on average similar, except for the extra education, right? So we have a policy that affected these people. There's no reason why these people born, days before the policy threshold, which is September 1st, 1957, is like a pretty arbitrary threshold, right? There's no reason why these people are going to be significantly different than that people, other than the extra education, right? So if that's true, by comparing health on people born here to people born here and asking, are there similar discontinuities in the health outcomes, we can say, okay, these discontinuities are for sure due to the discontinuities on education, right? So this is an identifying assumption that cannot be fully tested, because what we really want to say is that there are no confounding factors, right? These individuals on either side of the threshold are similar in all characteristics, observable and unobservable characteristics that you can think of. So there's no way we can fully test that. However, I want to say that genetic data are very useful in offering supporting evidence for that, right? Because what's the best data you can think of to test this kind of hypothesis is data that is, measures that are predetermined, they are objective, and they are determined at conceptions, and they don't change over the law, of course. So I would say that genetic data is as good as it gets for data that you can use to test this kind of assumption of whether people are similar on both sides of this policy threshold. So how do we do that? We do similar graphs that I showed you for education. We're going to ask, if we look at this data, at this, here BMI PGS, EA PGS, the different principal components of the genetic data, do we see any jumps, right? Do we see that at the policy threshold there are any evidence that these people from one side are different to the on the other side? And the answer is no, right? You can see that these graphs are pretty different than the education graphs. You don't see any evidence that there's any discontinuities. And we do kind of regressions that are like finer in date of birth, and we also kind of reject this hypothesis that they're not different. Okay, so another thing that could happen when you think of education and samples like the UK Biobank is that you could say, okay, the reform affected education, and we know that the UK Biobank sample is a selected sample of people who are more educated than the general population. So could it be the case that the reform itself, the education variation that the reform generated affected the probability of being in the sample, right? That would be problematic, because then you would say, okay, if the reform affected who is in our sample, who we see the data for, then that would automatically generate a violation of our identifying assumption, right? Luckily, there is a test that you can do. It's called the McCrary test, where we basically see the number of people at different birth dates, and we ask, do we see any change in, do we see that education affects the probability of whether you are in the sample or not? Do we see that, in one of the sites, education is, we see a larger sample than in other. And again, we don't see any evidence that that's the case. And the p-value there shows that also there's no evidence. Okay, so I think given that, we're like, okay, we're pretty comfortable. It seems like we have a natural experiment. The natural experiment has a first stage. It has an effect on the variable we are interested in. It has an effect on education. It seems like, we have a nice research design, the regression discontinuity design, and it seems like all the tests that we did to kind of try to see if the identifying assumption of the regression discontinuity design is valid, they seem to point that it is. Okay, yeah.
Yeah, I was actually wondering, so when you say that people are dropping out, that means that is the end of their schooling. There's no college, they don't go to college after that. 15. So
yeah, so the question in the survey is at which age you left full-time education, right? So yeah. Yeah, that's the question. So we, it seems like we have a nice design. So so what health outcomes are we gonna look at? So as I mentioned, we decided to focus on objective health outcomes because of the issue with the self-report, and on health aspects of health that we think could be affected by education, right? So we ended up with like 3 different dimensions of health: body size, lung function, and blood pressure. For each one of these dimensions, we actually had several measures in the UKB. So what we did to kind of avoid problems with multiple hypothesis testing is that we aggregated all these measures in 3 different indexes by each one of these dimensions. So we did that using the Anderson 2008 method. So for each index, we studied 2 types of outcome. First, the continuous index measure, and second, as I mentioned, an indicator whether the index is above a pre-specified threshold, which is the value we estimated the largest distributional effect in our previous analysis. So today I'm going to show you the threshold results only because, as we said, those are kind of our best powered results. All outcomes are going to be oriented, so a higher level means worse health. So I don't really need to talk much about this, but we're going to use polygenic scores. They are an average of a person's genotypes. As here is the polygenic score for individual I, and it's going to be a weighted average of like this number of alleles and the weight assigned. Then the weight comes from GWAS, from 2 GWAS, one for BMI and the other one for educational attainment. We couldn't find, at the time when we were working on this, sufficiently predicted GWAS for lung function and blood pressure. But because lung function and blood pressure are related to BMI, we're going to use the BMI polygenic score to look at those also. I'm going to show that, not surprisingly, it's not going to be as predictive as for BMI. It's also important to, again, keep in mind that these polygenic scores are associations. They're not capturing exclusively direct genetic effects. Alex, in his lecture yesterday, talked about all the ways there might be like indirect effects of these polygenic scores. Every time you're working on with these polygenic scores, it's very important to keep in mind that that's the case when you're interpreting your results. However, it is comforting that it's been shown that the EA-related hits are active in the brain and involved in neurodevelopment. So we think that, This EA score captures, among other things, some measure of cognitive ability. And the BMI scores are expressed in parts of the brain responsible for regulating appetite. So we think that there are at least some biological pathways that we can think about when looking at this. So one thing that is, I thought it was worth mentioning is that, again, we were really worried about power when writing this paper, right? So we wanted to do everything we could to increase the power of our estimates. One of the things that we did is that to notice that we were not using the full UK Biobank data, right? Because of our regression discontinuity design, we were actually only looking at those born within 10 years of our reform cutoff. So that kind of meant that we were only using half of the sample. So what we did with the other half is that we used that data to augment the published GWAS in order to increase the score's predictability. In other words, we couldn't use the UK Biobank data to generate the scores and in our analysis, because that would be overfitting. So what we did is that we kind of we got the data in the UKB that was not used in the analysis to augment the construction of our scores. And again, we did include that in the pre-analysis plan. So I'm going to start just showing you how those scores related to our outcome measures. Here is, I'm showing you the fraction above the threshold for body size, for our body size PGS. So remember, being above this threshold, it means worse health. So people here are very obese, in other words. And we have two sorts of estimates here. We have a linear estimates, which show that the BMI polygenic score explains about 5% of the variation in this body size measure. And we also kind of divided the BMI polygenic scores into three terciles. And what you can show here is that the people with the lowest genetic predisposition to having high BMI are about 10%, have a probability of being about 10% above the threshold, while those in the top tercile are about 30%. So there's about a 20 percentage point difference between those in the lowest, in the bottom tercile, and those in the top tercile. Okay, so when we kind of have our estimates, the question is going to be, what does education do to these differences, right? So does education decreases, increases, by how much? So this is kind of be the main research question. If this panel looks at the EA polygenic scores and again body size, as you can see, the correlation here is not as strong. We do see that those in the top of the EA polygenic scores have lower probabilities of having a high BMI index, but there's not a lot of slope there. Perhaps unsurprisingly, when we do the same thing for lung function, again, we see some slope, but the slope is not as strong, right? Because now we're using a polygenic scores for BMI to look at lung function, and there is some correlation, or some prediction, but not as strong as when you look at the trait itself. Same for blood pressure. Some, pretty much flatter lines there. So then we're gonna kind of move on to asking, okay, so what does education do to these differences? And the first step is to understand how does the effect of reform varies by those with different genetic makeup. So what I'm showing you here is that first step graph broken by BMI score terciles. And what you can see is that those in the top tercile of the BMI score distribution, that is those with the highest genetic predisposition of being obese, for example, were dropping out at higher rates than those in the bottom, right? The fraction staying in school until age 16 was lower for those with very high predisposition than those for very low predisposition. And what the reform does, because it's a compulsory schooling age reform, is that it kind of shrinks these differences to zero, right? It's saying for everyone who's born after this date, you must stay in school until age 16, right? So what it means is that it kind of washes out this pre-reform differences. So what we're going to see is that actually the effect of the reform is going to be larger for those at the top tercile of the score distribution than those at the bottom. Yes?
No, I had two clarifying questions.
Mm-hmm.
One, is the initial difference per reform, do you think is that because there's a true effect of BMI on true population of education, or is speaking of something about family environment?
Oh yeah, I think for sure it's picking up something about family environment, neighborhood environment. Yeah. I'm not, again, I think the point here that these polygenic scores are not, our associations are super valid here, yes. Mm-hmm.
And the second question, maybe you already mentioned it, maybe we missed it, but why is the node 1?
Because again, there is this summer, people who were born in the summer,
Okay.
even after the reform, they could, so
even
when...
Later on, like at 65?
Yeah, so you still have like, you have that, if I drop everyone who's born in the summer, this would go to 1.
Okay.
But there's pretty good, the compliance with the policy was pretty good. It's just we have this seasonality because of people who were born in summer months. And this has been documented in other data sets also, yeah. Okay. So if we do the same thing using the EA polygenic scores, unsurprisingly, these differences are even higher, right? Because again, now we are relating EA score to an education variable, right? So you can see here that people in the bottom tercile of the EA polygenic score are much more likely to be dropping out before age 16, right? And again, kind of like these things shrink significantly after the reform. So what the reform is doing is that it's bringing these people in the tail of the PGS distribution. It's forcing them to stay in school longer. Okay, it's forcing. The effect here is also significant, but the effect for the bottom is just going to be much larger. Okay, so in our empirical strategy, we're going to ask, how does the effect of this extra education, how does that affect health, right? We're gonna we're gonna have two different empirical specifications to look at that. One linear specification where we're gonna regress our health outcomes on the score that we're interested, on the variable, whether you stay in school until age 16, and on the interaction, right? And we are interested in this coefficient, the G by E coefficient. We're gonna instrument, because whether you stay in school until age 16 is an endogenous variable, right? It's a choice variable. We're gonna instrument this with whether you were born after the reform, with an indicator of whether you were born after September 1st, 1957. We're also gonna add all the first 15 principal components to control for ancestry and stratification, and we are gonna include quadratic polynomials in exact date of birth, and that's kind of part of the regression discontinuity design, where we have these trends before and after the reform, and we wanna control for them. In the paper, we show that all the results are robust to kind of linear trends also. So we also have controlling for predetermined variables such as gender, age squared, country of birth, and calendar month of birth, which to deal with the seasonality that I mentioned. So when showing you these results, I'm going to be plotting beta 1s plus beta 3 hat against S1 to show you kind of the linear projection of how the effects of education on health are related to the scores. Sorry about that. I have no idea where this is coming from. Okay. This didn't go. The other empirical strategy we're going to use is a less parametric empirical strategy. We're going to basically divide the sample in three, right? In three terciles, bottom, middle, and top tercile of the PGS. And we're going to kind of I'm going to show you the bar graphs against the median PGS for the tercile. So again, just a linear specification and a less parametric one. So here are the effects for body size. So the way to read this effect is that, again, this is the BMI PGS. This is the effect on the fraction above the threshold of body size, right? And this is the linear specification, and this is the tercile specification. So what this shows is that for people with low BMI score, we basically find no significant effect. It's like a small positive effect, but it's like the standard errors are huge, right? However, and we moved in the BMI score distribution, we start seeing very large negative interactions here. So what this is saying is that for people with high predisposition to obesity, the effects of education were much larger than for people with low genetic predisposition to obesity. The difference here is about 13 percentage points. If you remember when I talked about the baseline differences, they were about 20 percentage points across like these two groups, the top and the bottom tercile. So what this means is that education is closing the gap in the association between, score and body size from 20 percentage points to 6 percentage points. So it's a pretty strong effect in the compression of this association between scores and outcomes. If we do the same with the EA polygenic score, we don't find a significant effect. So you can see that kind of here's the p-value of the linear interaction. This is a very small p-value. It survives a number of corrections for multiple hypothesis testing, but not the same. We don't see the same G/E interaction with the EA. Yes.
So I think I missed this. What gap is education filling on the left side but not the right side when we use the EA score?
So what gap? So the gap we had before between this group and this group was about 20 percentage points, and it's filling 13 percentage points of those 20. Okay, here I don't even know, but the gap wasn't as big to begin with, but we're not really, those are not statistically significant, like they're not different. We can't say that they're different. So the answer is we don't have any evidence that they're closing any gaps at all. So for lung function, we see a similar result, that the effect is concentrated among people with higher, the effect on body size is concentrating on lung function, is concentrated among people with larger scores from BMI. But the significance of this value is much larger, right? The p-value is much larger. So what it means is that when we kind of do our Bonferroni corrections is that this result doesn't really survive those types of corrections. And again, not much of we don't have any evidence for interactions with the EA score. Yes.
Just another question. So suppose you did the same analysis but without the intervention. So you could do like two ways. One, just interacting with actual user schooling and there was no
right.
Control, or just interacting with the EA PGS.
Right. What this looks like. We never did, like people have asked us before, but I don't think we ever did the OLS version of this, did we?
I don't think so.
Yeah, I'm pretty sure we would find. No?
There are papers that do it, right?
Right,
like there's
right.
some Swedish group that emails me occasionally about it.
It's true.
But yeah, so there are groups that do it, and I think they are finding similar kinds of interactions in the cross section.
Yeah, I would expect.
Polygenic scores? That would be interesting to know There's an interaction between the BMI PGS and the EA PGS in similar ways.
But interactions between the two scores? I don't understand what that means.
So the idea here,
Hmm.
right, is that when you increase education, you decrease the effect of the BMI, genetic propensity for BMI on the actual health. It would be interesting to know if you look at individuals with a high propensity for education, they also have a
But a low.
effect of the BMI, the propensity. You know what I'm saying? This is G by G. I'm just curious.
The correlation between the two scores is minus 0.24 or something, right? So they do go
But I'm talking
about the interaction on with the health. Yeah,
So that would be interesting.
yeah, yeah. I don't think we've done that. Yeah.
So they should be operating similar mechanisms.
Mechanisms, like, no. Okay, blood pressure. You know, maybe not surprisingly, since we didn't see much of a prediction power to begin with, we don't really see any effects there. So as I mentioned, this additional education reduced the gap in unhealthy body size between the top and the bottom terciles from 20 to 6 percentage points. We find no evidence of a similar interaction between education, between health and the EA polygenic scores. And then there's the question, so why? Why do we see that those with higher genetic predisposition to obesity have larger improvements? It might be that they might make larger adjustments to their health behaviors when they got the extra education. It might be that just a given adjustment translates into like larger health improvements, right? We're not terribly, with the data we have in the UK Biobank, it's not great to look at that, but we tried. We see that the reform on average led to higher qualifications, increases in income in middle age. These people who were affected by the reform had better jobs that were like higher socioeconomic status, and they ate diets with less fat and less saturated fat. But when we did the interaction with the BMI polygenic scores, we do not find that the group for which we find the largest effects on health had the largest effects on these mediators, right? So we don't really have a lot of evidence that we see larger adjustments for this group. This should kind of be taken with a grain of salt because there are limitations to this data. Some of this, the diet data is only for a subsample sample. Some of this data is self-reported. It's a single snapshot in time. So there are things that it's not the ideal way to test this. Yeah.
Yeah, I know you said it was a subset sample. I think I might have missed it when you said it. But how many people were actually in that 20-year window of birthdays?
It's about half the sample.
Of the whole of UKB?
Yeah, but here what I mean is that they didn't ask diet for everyone. So not everyone answered the diet. Only people who are still employed have the occupation. So there are like things with this data that make them not ideal to testing these things. So just to conclude this part of the presentation, we see that additional education reduced this association between genetics of genetic risk of obesity and actual body size. Kind of this challenge, these notions that there is a genetic determinism. We kind of in this case show that social policy can mitigate these differences that come from genetic risks. However, we also want to point out that that we see kind of these results as kind of a data point, and it would be kind of very important to kind of understand whether they replicate, whether they're generalizable to other contexts as kind of the availability of data grows and other scores become predictive enough. So we think that this kind of can be a very interesting research agenda moving forward, thinking of ways where you can find natural experiments to look at causal estimates and coupling them with like polygenic scores in samples that are large enough. So I can't stress that enough. Like to do like GBI studies successfully, you do need samples that are large, right? As we talked in the beginning of the talk, one of the main obstacles is low power. So Patrick, when we were writing our pre-analysis plan, he did some power calculations based on some preliminary data that we had, and he came up with this number that for interactions in the magnitude that we had with a fourth of the sample, we needed samples of at least 10,000 or even 100,000 to get adequate power to get at this GBI effects, right? And this is in part because these GBI effects are much smaller than direct effects or than effects on correlation, right? When we're trying to get a causation, effects are going to be smaller, and the interaction effects are going to be smaller than the main effects. So I think this is a very promising area of research, but you really need to be kind of careful when picking where to invest your time, right? You really need to think about power calculations that take these things into account, right? You really need to be thinking, when I get my source of variation, how much of the sample is being affected, right? In our case, it was 14%. If you have a source of variation that's affecting all the sample, you're going to have a more powerful experiment, right? So there are many things to consider when deciding, is this a promising area to try to estimate GBI effects. Yeah, someone had a question.
Do you have a sense for how this might change with the EA3 score, which is maybe possible?
So a lot of the increase in predicted power came from the UKB, right? Is that right? That's right, right?
And 23andMe, that was a big.
So we're using for the SES stuff, we're using so I'm going to talk about a bit of stuff that we've been doing now, where we're looking at SES outcomes, socioeconomic status, as an outcome, and that's using the EA3. I don't think we revisited the health outcomes with EA3, but also I don't think the change was big enough to make a difference in the results, right?
Not in the results, but in the power calculations?
Yeah, I mean, yeah, but I think the main, I don't know, have you redone the power calculations with the EA3?
But like the R squared going from EA2 to EA3 like increased predictive power by perhaps 50% or something like that. And these numbers here are like order of magnitude differences. And so I mean I think the same point will apply even with the more powerful polygenic scores, that interactions tend to be much smaller than the direct effect.
Yeah. Okay. So in the next few minutes that I have, I wanted to talk a bit about some ongoing work that you've been doing, like where we think we're gonna go from here. So we're thinking of we're using kind of the same data and natural experiments to look at how education and for interactions between education and the EA polygenic score in socioeconomic status in middle age. And there we do find some interesting interactions. So just to give you kind of a sense of what our results look like, this is kind of the outcome here is occupational SES. It's kind of this idea that your occupations can be rated into how prestigious they are. So a higher number here means like more prestigious occupations. Here is school leaving age, so it's kind of similar to the Edu16 variable, a higher school leaving age instrumented by the reform means that you get better jobs in middle age. Okay? So interestingly, when we do the interaction with the EA score, we find that those with large EA score see larger gains, right? We see here that, schooling is good for cool jobs. Having a larger score is also increases your probability of having having good jobs, but the interaction between schooling and education is larger for those with higher scores. So it's kind of interesting to think that this actually goes against a bit what not against, but we saw in the health example that education shrinked differences across PGSs, right? Here the interaction is positive, meaning that it's those with high scores who were more likely to have better occupations to begin with, which gained the most from the extra education. So here, I don't have the exact numbers, but here if we did like the same bar graphs, education would be increasing those differences between bottom and top of the distribution. Yeah, that was a question.
Sorry, I just want to know, what are the local SES controls? Is it location controls?
Well, I'm going to talk about, it's not here. Yeah, I'm going to talk about in the third, and it's coming. Yeah. So here I'm doing exactly the same specification that I talked in the education, right? In the health examples, here we have like principal components. We have those same predetermined controls and controlling for trends in date of birth. And by the way, if we look at other SES outcomes such as income, we see kind of the same pattern. Okay, so I just chose occupation here. So one thing that's, that we saw yesterday in Alex's lectures is that, okay, how do we think about these interactions, right? How do we think about these scores, right? They're not only capturing kind of a direct effect from your genes to your outcome, but there's also kind of a lot of indirect effects, right? So he mentioned parental the genes of your parents are actually an omitted variable here, right? So if your parents have higher scores, they're probably gonna pass to you higher scores, and they're also gonna be able, for example, to provide you with a better growing environment, which also will affect your economic status later in life. So what we're doing now is that we are trying to think about how much of these interactions with genetics can be explained by differences in the environments you were born in. There's a growing literature showing that where you're born, where you grow up, matters a lot for your socioeconomic status in later in life. And the UK Biobank actually has respondents' exact coordinates of birth. So we know within the UK exactly where they were born. So what we are working now is to match these coordinates of birth to measures of local SES at birth. So we're trying to get measures that are as local as we can get to ask the question, are the interactions that we're seeing with genetics, can they be in part explained by these indirect channels, right? By the fact that you also have interactions of the reform with with the level of development of where you were born, where you grew up. So we do that. We kind of have some preliminary measures where we linked these geographical coordinates of birth to the fraction at the local authority level, which is like the finer level that we could get at this point. So we get the fraction of people with different occupations, like prestigious occupations, and the fraction of people at different educational levels at the time of when the participant was born. So the first question we asked is the effect of the ROS on education larger or smaller for individuals from disadvantaged areas? And maybe, not surprisingly, we find that similar to when we broke by PGS, we find that the effect is larger for those who were those who were born at disadvantaged areas, just because those are the kids that were most likely to drop out from school earlier, right? And then the next question that we wanna think is like, okay, once we control for these differences, once we interact these local environmental measures with the effect, does the interaction, the G/E effect that we see, does it go down, right? If we think that the polygenic scores have a direct and an indirect component to it, once we try to add measures that will capture this indirect component, this indirect channel, how much do we see that what's the direct component that survives? And for now, it seems like it's not much. So these are the local SES controls that you asked. If we look at the interaction here, it's like we see like if anything it goes up, but it's not, it's very, very similar. We're thinking of whether, we're trying to get, these controls are predictive of later life SES, but we're trying to get at like, okay, maybe they're just not good enough proxies of environment. So I think going forward, what we want to do is try to get, the best local level proxies of birth environment we can get, and redo this and see, okay, how much, these measures are going to be noisier, how noisier they are, but how much of this can we explain? And yeah, the first here and then.
I wonder how much of a difference it made to control for all 15 principal components versus 10 versus 5, for example, and how is that related to your measures of local SES controls that are also geographical in nature?
Right. Yeah, so we are controlling for the PCs and the interaction. I think the main difference is that the principal components are gonna be a variation of like ancestry versus like really SES. What do you think? I would think that the interaction with SES. I know these are all related. So maybe what you're saying is that, you wouldn't expect much of an interaction with these SES measures if a lot of it is already captured by the PCs. But we do see that they are significant. Like I don't show them because there are a lot, but they're definitely affecting the, above and beyond the PC interaction. So.
How do we know you're not underestimated in a sense? I don't know.
Yeah.
Underestimating the environmental things? I think we know that we are underestimating.
Right.
Which is, I think, the point that Silvia was.
But could you see the P control for PCs, for example, and see the differences?
But I think we're still, when thinking about these biases, right, on the scores and how do we interpret them, I think we still I don't think dropping the PCs is the answer, because we're still worried about ancestry and certification, right? So we still wanna control that for that. I think the idea of adding this local control is to, if anything, try to get at these other types of biases that could be potentially driving this interaction. You don't like the bias, saying it bias? Okay. Well, we'll talk later. You made a terrible face. Okay.
So Matt told us that for these kinds of controls, we need to interact them with the treatment. So
we do, yeah. This is control, and control interacted with the treatment. And I was so happy that we actually, before he said that, we already had demeaned this, because we noticed that was important. Demeaning this is very important also. Yeah. Yes.
Do you have other measures of early life apart from regional birth?
Yeah, so that's one of the things that is like not so great about the UK Biobank. They have a lot of good health contemporaneous measures. They don't have a lot of good SES measures, and they don't even, going back. We have things like whether your mother breastfed you, what else do we have? Like some things are very soft that we think people, it's self-reported, and we just don't think they're great measures. So that's why we decided to, we have coordinates of birth if we have, and there's kind of some variation at local. No, we have when they died, if they died. we have some health conditions. It's really not great. Yeah. There.
So every observation have the same weight within your group?
No, we do like kernel weights where we are giving like larger weights to people close to the policy.
Because it sounds surprising to me, like in a RDD framework, you have such a big window, because I will worry about other policy changes or other, structural transformation.
Yeah. Yeah, we do the same. So yeah, that's the thing. That's interesting with RDD, right?
Particularly if there is 1973 restructuration of health system in the country, so that could also affect the in the same timing you can have other policy effects. Yeah,
but you think like this is not the timing. This is not the time period, right? It is the date of birth. If you think that something, some policy, some restructuration affected people of different ages similarly, this wouldn't be a problem here. But I take your point that 10 years is a pretty large window, and in the paper we do we do kind of some robustus with 5 and 3 years. But you're always with these RDD designs, you're always trading off bias and power, right? And because we were so worried about power, and we thought that when we looked at our outcomes, the trends were actually fitting pretty well, we decided to make the 10-year our main specifications. That's why. That's what drove our decision. Yeah, you had a question?
Actually, it's very related about policies around the same time that may confound that.
Yeah, I think you would have to be a policy that affected that cohort only, and we don't know of any. There's actually, we're not the first ones to use this reform. There's actually a paper at the AR that uses this reform. We're the first ones to kind of use it together with genetic data.
Nice.
Yeah.
It's a very small point, but I think previous papers in this literature excluded people from Scotland because the policy applied in 1976, not in 1972.
Yeah, so I don't think that's true. So I was actually visiting the UK when we wrote this paper, and I talked to some Scottish, and we looked for the actual law, and then there was someone from Bath who actually followed up with a paper. Yes, talking about, so I think it's a misconception that it was the wrong. That was the wrong time.
I think the Dixon paper says that it has an effect, but still that the reform might have applied in 1976, but maybe there's some.
Right. But even if it was okay, so you're saying 70, but as long as it affected the same cohort, that's what all we care about, right? That's what we care about. It was a different time but it was the same cohort. So here it's all about what's the first cohort that was affected. Right.
But I wonder how if there's some non-compliance for the Scottish people, even if that discontinuity is still at that same point for the Scottish people.
No.
I sort of wonder how that might bias.
Yeah, I mean Scotland is not a huge. I think it's less than 10%. We probably, I don't know if we ever drop them. I don't think it would make a huge difference.
I think when we decided to add them, I'd have to double check. I feel like we actually checked just Scotland to make sure that we saw the discontinuity.
Right.
And you see it. It's pretty clean. Even
we're letting the data tell us whether it's there or not. Yeah, that's a good point. Yeah. Yeah, but I agree that there was some controversy. It's it's there, it's there. Does that apply to Scotland? Not, because some of the papers exclude Scotland. Yeah. Yeah.
So you said you were looking at local SES controls for some kind of contextual job stratification, right? Professional or percentage of people in a certain professional band.
Right. So the idea is that we see this G/E interactions, right? And but we also talked yesterday in Alex's lectures about how these polygenic scores are associations and how they include effects that are not only direct genetic effects, right? And one of these indirect effects could be effects of the environment, right? If, for example, your parents have better have better scores and they can provide you with a better environment, that's gonna affect your SES. So what we're saying is that, okay, what happens to this genetic interactions once we do our best to control for this changes, these differences in birth environment. Yeah.
Yeah, I mean, I guess I was just going to say that, like, spatially, I don't know how fine-grained it is. If you said you had local authority, you can get, there are like 3 censuses in there, right? So you can link to
So the data that we get is called the Vision of Britain. It's a data set that basically uses, it feeds from the census.
Sure.
So this, right. So it's just, we got the data from there because they had this nice, we could just download, like, by the local authority, like these things, but it comes from the census. I think we're trying to add more measures, and maybe some of the other measures come from other surveys, but I know that a lot of it comes from the census.
Yeah, I guess also you can have some kind of spatial weights between them, like, depending on how close, if there are certain areas which exert a bigger effect from local SES or something like that. I don't know, maybe we can just talk about later. But
Right.
Yeah. Lots of
We haven't spent a lot of time looking at the SES. I think that's something I want to do.
Yeah, sure.
Looking at the SES interaction. Here I'm just saying, okay, I'm adding this and this not. But actually, I think there's some interesting stuff to just look at the SES interactions themselves.
Yeah.
And how do you think about those, and how do they compare to, all the like the chatty kind of estimates and things like that. I think that would be
We should
cool.
also maybe highlight that these results are very, very
Recent.
preliminary.
Very preliminary,
Like,
yes.
so like we've coded up the variables independently, and our answers don't even match yet.
Yeah.
And so
And then I was like, oh, but I want to talk about them anyway.
But yeah, so the goal is mostly just
Yeah.
that these are things that we're thinking about, to illustrate some of this stuff.
For sure, it might change radically. Yes. Yeah.
Just one of the
Yeah.
Not for this one, just for the BMI stuff.
Yeah. So maybe in 5 years we'll have a paper about this. That's how fast we move.
You should mention what the award was that this paper got. The UK Biobank. You want to explain?
Oh yeah, he's the UK.
Yeah, well, I mean, I feel,
Patrick.
okay, so it's there was an award for the UK Biobank Early Career Researcher of the Year award, and so I sent this paper in as part of that competition, and they announced today that we got that award. But I feel a bit funny because